Hello and welcome. I'm going to show you guys today how to navigate through the laboratory manual. The laboratory manual again is free and you have it here for your use. If you're in class, you'll print it out and bring it to class. Here we're online, so you've got access to it here 24/7 basically. I would suggest you download this into your computer. That way you have again 24/7 access to it in case the website goes down or something or another happens. You have that there. Now here we'll start off with first histology. When you click on histology, basically this will open up. So here what we have now is you can see the first page of your laboratory manual. This is the table of contents you can think of. It goes through and it breaks everything down by modules. We have it by lab practicums. So here we have 6 lab practicals, and you can see all the material here for each of those 6 practicals. Coming all the way from histology, skeletal system, muscular system, nervous system, viscera, down to cardiovascular system. Now, here we'll start off with first histology. Now, in histology, you guys have epithelial tissue first. After epithelial tissue, you'll move on to connective tissue. Connective tissue, you can see we have 2 parts here. Part 1 is connective tissue proper, and part 2 is going to be connective tissue as well. Part 2, cartilage, bone, and blood. And then last we have muscle, nervous tissue, and integument. So I'll show you how to navigate through here this first part. Now here we'll move down, and here you can see you've got epithelial tissue. So like it says here, these features you have to ID on all epithelia. First is the apical surface. In the lecture we talked about the apical surface. We can see here, if we want to look at the apical surface, we can identify that apical surface as being the upper surface, the surface that's going to be containing either cilia, it could be smooth and slick, or we said it could have microvilli there. Next then it will be basal surface, right? And then at the basal surface you can also identify the basement membrane. Next then here we have the lumen. So here we want to be able to identify these features on epithelial tissue. Now what we'll do is we'll move down. We'll skip these first 2 slides. Okay? We'll skip these first 2 slides and we'll move down all the way to here. So when we move down here, we can appreciate now this pointer pointing at epithelial tissue. More specifically speaking, you can see it's pointing to a specific region on our epithelial tissue. Now, same thing we have happening here in the next image as well. Now here in this first image you could see this is the pointer, and here we've got our epithelial tissue, right? Here would be basically, we said it's a lining tissue, so here you can have the surface, basically is going to be what you see here. It could be either the outer surface or it could be the surface into a cavity or whatever it may be there, whatever we're talking about. We're going to get down to it, pin this down, and see what we're looking with, or see what we're looking at and dealing with. So here we've got now first just one row of cells. These cells you can see are supported by muscle and connective tissue down here. Now these cells are squished and flattened cells, right? They're not cube-shaped cells. So here now you can see one layer, simple epithelia, which not cuboidal, not columnar, but squamous. Simple squamous epithelia is what we're looking at here. Now this pointer, you see here, it's pointing to the basal surface, where that basement membrane would be. So if I was to point down here and ask you what surface are you looking at, or what structures can be found here, and you have to look at your options and see reticular lamina, basal lamina we talked about there, and basement membrane we said is making that up. So you have to be able to identify all of that being here at the basement membrane. And then here you've got the apical surface, which you can see is being pointed at in this first picture. I'm sorry, in this picture right after this picture here. So on page 3, the second image here on the bottom, the pointer is pointed to the apical surface. So here you've got your simple squamous epithelial cell. Okay, here's another cell right next to it, another cell right next to it, and then there'd be another one here and going all the way down, and the same thing all the way around this way. Then here we have connective tissue and then muscle tissue or whatever tissue might be down there. So this pointer is pointed to the apical surface of this cell. So Here you can see now coming all the way back up, apical surface, basal surface, and there you have the basement membrane. Now the lumen. The lumen is basically the central cavity, the space. So here, okay, the lumen would be basically in this space right in here. Right in here, the lumen would be right up in here. Okay, this here we could see we've got these tubules right in here. Okay, right in the center would be the lumen. Okay, if I was to cut it here and show you just half of that picture, right, the lumen then you would say is basically on the top. So same thing here, the center, the space in here would be the lumen. That apical surface is pointed towards the lumen. It's pointed towards the lumen. So the lumen would be the space in here. We'll review those terms as we progress as well. So here then let's start. We've got apical surface, basement membrane, and lumen understood. Now, first slides you're going to be identifying here are going to be in relation to simple squamous epithelia. Okay, first you have to be able to identify it. If I show you simple squamous epithelium, you have to be able to say this is simple squamous epithelia. You have to be able to talk about the locations of where the simple squamous epithelia is found, as we've mentioned in the lecture. You have to be able to identify its functions, as we identified there as well. Now, we had mesothelium and endothelium. Mesothelium and endothelium, we said, were special simple squamous epithelia. They're still simple squamous epithelia. Mesothelium, we said, is going to be found making up the serous membranes. Okay, so mesothelium is this first slide you see here. Mesothelium. If I want to show you mesothelium, I'll always show it to you from here. Here I won't ask you for apical surface or basal surface because all you can see is apical surface. So here what you're doing is you're looking at basically that cell. Okay, let's say here my phone is the simple squamous epithelial cell. Okay, we'll show it to you guys here. Here you can see it's my wallet. I keep my wallet on my phone, so it's right in this case. So you can think of that as a nucleus. So here is the simple squamous epithelial cell. What you're doing is you're looking at it from the top. Okay, whenever you look at mesothelium and endothelium, you will always see them from the top like this. Okay, normally speaking, in regular simple squamous epithelia, as we've seen in the lungs and these other places, you're going to be looking at it from anterior view versus superior view. Okay, you'll be looking at it from the front, just like you're looking at it from there. You can appreciate the nucleus, okay, and you can see everything there. So from here you can see the nucleus, but it's going to be basically just an image like this. Here, it's more of a 3D image. So first slide is mesothelium. Second slide, endothelium. Best way to distinguish them is the color of the stain. Okay, same thing here, superior view. So, simple squamous epithelium, identification, location, functions. Mesothelium, endothelium, and then last is Bowman's capsule of the kidney. So let's move down here. Okay, we went through this slide here. Simple squamous epithelia, it's pointed to the basal surface. Simple squamous epithelium pointed to the apical surface. Now here we're going to identify simple squamous epithelia as it says in Bowman's capsule of the kidney. Now, Bowman's capsule is this capsule I told you about, glomerular capsule, Bowman's capsule that's surrounding that glomerulus. So right in here, this is a picture of a nephron. But at the center of the nephron that we're looking at here is, you can see the glomerulus right in here, and surrounding it is that capsule, Bowman's capsule or the glomerular capsule. Okay, this is the renal corpuscle, more specifically speaking, but we'll see that as we progress. Now here then you've got the PCT, proximal convoluted tubule, and it's going to make its way then to the loop of Henle as I drew, and then the distal convoluted tubule, then the collecting duct. So here you can see, I told you you're going to have simple squamous epithelium here on the side, on the outside, and there you go. Right here, you can see right inside of here, right inside of here. Same thing here. It's all simple squamous epithelial cells all around here. I told you you are also going to have some here in the glomerulus, but here it's hard to find. I'm not going to ask you about them. In the lecture, I can ask you, where else are they found, and you could identify them also as the glomerulus there as well, also in the glomerulus. So Bowman's capsule and the glomerulus. And also mentioned to you guys there, inside the lungs, right, as well. So make sure you've got all that understood there. And read thoroughly and get that thoroughly understood. Next, then, let's move down. So next we have here simple cuboidal epithelium. Same thing with simple cuboidal. You have to be able to identify it. You have to know the locations. You have to know the functions. And now here I'll show it to you from a renal tubule of the kidney and be able to identify it there and see what you have there. And then the thyroid follicle of the thyroid. So let's move down. So right inside of here, you can see first, we took care of this slide. Now right in here, you can see this picture right here. This is now the tubules. So if we continue with the tubules, the PCT, right, the DCT, the loop of Henle, now you're going to see some of those segments are going to look just like we're seeing here. Now when we zoom in on these cells there, you can see here cuboidal cells, cuboidal cells, cuboidal cells, cuboidal cells, simple cuboidal cells. Down here is where the connective tissue is. So down here is where the basal surface is at, right? All down here. Apical surface would be right in here, because right in here is the lumen. Very good, the lumen. And here's the lumen, here's the lumen, here's the lumen, here's the lumen. Okay, so that's what we're seeing here. Next, then, here's the thyroid follicle of the thyroid. You can see each of these follicles are made up of simple cuboidal epithelial cells, simple cuboidal epithelial cells, simple cuboidal epithelial cells, simple cuboidal epithelial cells. So there's one follicle. Look at how many of these simple cuboidal epithelial cells it takes to make up one follicle. Here's another one follicle. Again, look at how many simple cuboidal epithelial cells it takes to make up one follicle. Another follicle, right? Another follicle made up of simple cuboidal epithelial, simple cuboidal epithelial, simple cuboidal epithelial, simple cuboidal epithelial, simple cuboidal epithelial. We're going to look at them in greater detail when we get to the thyroid gland. Next, then, we have simple columnar. So simple columnar, okay, identification, locations, and then the small intestine, you're going to look at it. In the small intestine, you're going to see goblet cells and brush border microvilli. And then also you're going to look at it as ciliated simple columnar epithelium from the fallopian tubes, the oviducts. Again, you'll see the cilia there also. So simple columnar epithelia first, you can see digestive tract right in here. Simple columnar, another 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 one next to it, another one next to it, another. Simple because it's just one row. What is this in here? Connective tissue, separating this row because this is a villi. So the apical surface would be up here, basal surface down here for this row of cells. Apical surface up here for this row of cells, basal surface down here. So we move to the next slide. Same thing, digestive system, you could see the villi. Here you can actually see the goblet cells. And here then we can also see, we'll zoom in this area, you can see the brush border. All that dense microvilli, all right inside of here, brush border, and then goblet cell. Here's the fallopian tube, the oviduct. Now some of these cells are not ciliated. Some of these cells in here are ciliated, like right in here. Non-ciliated because they're going to be nourishing the cell as it passes through the ova, and the ciliated cells, they're making sure it moves forward. And then some more ciliated in here, non-ciliated, some more ciliated, okay, coming around and then looking around here, non-ciliated, non-ciliated, and then a whole bunch of ciliated, and then non-ciliated, ciliated, non-ciliated, ciliated, non-ciliated, ciliated, okay? And here you can see how that row of cells comes around this way. Again, simple columnar. Simple columnar, simple columnar, simple columnar, simple columnar. And here's the connective tissue providing support. All of this connective tissue coming up into here, connective tissue providing support. Coming around up into here, providing support. So here then we have pseudostratified, ciliated pseudostratified columnar. The cilia you see right up in here. Okay, identification again, location, functions, goblet cells. Here's a nice goblet cell right inside of here. Okay, the best one I think if I ask will be something like this, right in here. And then here you can see connective tissue right down inside of here. Next, then you have stratified squamous epithelium, identification, locations, functions, so the goblet cells we had over there, stratified squamous. So identification, location, functions, and then here you have two types: non-keratinized stratified squamous epithelium is what we're seeing here, right? Non-keratinized because you don't see any dead cells. In other words, we're going to see a whole bunch of dead cells up here. You can see all these cells up here have nuclei. A whole bunch of them have nuclei, right? Nuclei stratified again because many layers. Right down here is the connective tissue, and then from up here or up in here is where the epithelium starts. And we said these are going to be, the basal cells will be squamous, they're going to be cuboidal in shape, and then flattening out basically as you progress superiorly. So non-keratinized, and then here you can see stratified squamous, keratinized. So keratinized, stratified squamous epithelium, and this is from epidermis. So this again could be from vagina, mouth, moist areas as we said. So stratified squamous, keratinized from the epidermis. You can see a whole bunch of rows of flat and dead cells right outside of here. All flat dead cells right outside of here. Again, all connective tissue and basically some muscle tissue we can see down there as well. And here you've got all basically epithelial tissue, epithelial tissue, epithelial tissue, all epithelial tissue, all the way up to there. Next, then you have transitional epithelium. So here, transitional epithelium, you could see the locations. So here you could see the dome-shaped cells, right? This is the row of cells. So the dome-shaped cells on the top, as we mentioned, depending on the degree of stretch. And then right down here is all connective tissue, and then muscle tissue we'll have down there as well. So basal surface would be down here. Apical surface would be up here. And then right in here would be the lumen, the central space. Next then is connective tissue. So you're going to do the same thing with connective tissue. Okay. First is embryonic or mesenchyme connective tissue. You have to be able to identify it, know the locations, and know functions in regards to it. And you can see that right in here. We'll zoom in on this picture, and that's what you're seeing here. So again, mesenchyme, embryonic tissue. From the lecture, you'll see what we talk about in relation to this. Okay, next then here we've got areolar connective tissue. Same thing here, identification, locations, functions. And here you have to identify elastic fibers and collagen fibers. Elastic fibers and collagen fibers. The elastic fibers are the thin fibers you see here. And then the collagen fibers are the thick. So as we were saying, we could see all the collagen fibers, the thicker fibers, all inside of here. All coming down here, here, right inside of here, all up in here, all up in here, versus the elastic, the thinner fibers you see right here. Kind of more in the purplish, a darker purple, versus kind of like a red, the collagen, thicker ones. And then also here you can appreciate the fibroblast nuclei. Fibroblast nuclei. Fibroblast nuclei. Fibroblast nuclei. Fibroblast nuclei. Fibroblast nuclei. So this is all connective tissue. So this is when the lab requires you to do connective tissue. This is when you'll start connective tissue. This second part here from page 9, and second part of histology, which is basically connective tissue, and you'll move in from here. So elastic fibers, collagen fibers, fibroblast nuclei. Next, and moving down, you have adipose. Okay, so here you can see when we talk adipose, then here we've got adipose. Now, when we talk adipose, same thing here: identification, locations, functions, adipocytes. Look at all these adipocytes. Here's one adipocyte. Here's another adipocyte, another fat cell, adipocyte, also known as, right? The fat cells are known as adipocytes. Fat cell, another adipocyte, adipocyte. This is a blood vessel. Adipocyte. Look at all these fat cells. Now here is the nucleus. The nucleus is pushed off to the side. Look at here. The nucleus is pushed off to the side. Here you can see the nucleus is pushed off to the side. Now why is the nucleus pushed off to the side? The nucleus is pushed off to the side because inside is the lipid drop. The fat is inside of that fat cell. So the fat comes in and just basically takes over and pushes everything off to the side. Right? Cytoplasm would be inside. That cytoplasm is made up of water, so lipids and waters are mixing, so it pushes everything off and takes over that space right inside of there. And that's why you can see that nucleus is kind of squished off to the side there. Next thing you have the lipid drop, so that's inside of here, inside the adipocyte. Okay, the nucleus is out off to the periphery. I like to call these diamond-shaped cells, right? If you look at a diamond ring-shaped cells, right, a diamond ring, right, the diamond would be right on top, so it kind of looks like the diamond on top. If you were to turn this ring around, it looks like a diamond on top of the ring. Next in here we have reticular connective tissue. Now here, reticular connective tissue, identification, locations, functions. Okay, trabeculae now you can see here. Here's some trabeculae, here's another trabeculae, here's another trabeculae, here's more trabeculae, here's more trabeculae. They're basically septa. They come in and they divide the organ up. So that's what you're seeing here. Some more in here and here. So we also want to be able to identify the reticular fibers. Now from this zoomed out view, you can't see the reticular fibers, so what we'll do is we'll zoom in on this tissue. When we zoom in here, you can see the reticular fibers. Reticular fibers. Fibers, more fibers, more fibers, more fibers, more fibers, more fibers. All reticular fibers. Okay, here you can't see the trabeculae. The trabeculae you can see from here. Next thing you have dense, regular, white, fibrous. Dense, regular, white, fibrous connective tissue. Identification, locations, functions. And now here you have to identify collagen fibers. So you can see right in here a whole bunch of collagen fibers. All collagen fibers. Collagen fibers, collagen fibers, collagen fibers. Here the fibers have been stretched out, and here's some space that gets created in between the fibers. And then here you also have fibroblast nuclei. Fibroblast nuclei, fibroblast nuclei, a whole bunch of nuclei there as well. Nuclei, 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 fibroblast nuclei, 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 nuclei. All fibroblast nuclei, fibroblast nuclei, fibroblast nuclei, nuclei. More nuclei, 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 nuclei. And then collagen fibers, collagen fibers, collagen fibers. Nice parallel arrangement of collagen fibers. Compared to dense, irregular. Dense, irregular, white fibrous connective tissue you can see. So you have to be able to identify it. Locations, functions, collagen fibers here are all haphazard. And that's going to be because of the function. It benefits the function. So here you can see these collagen fibers are not all nice and parallel arranged like we saw here in dense, regular. So dense, irregular, look how they're all just arranged everywhere. And here you can see fibroblast nuclei, fibroblast nuclei, fibroblast nuclei, again fibroblast nuclei, more, everywhere. All these purple staining fibroblast nuclei. Next then, I want you to add in there slide number 7. So here you can put slide number 7 and add elastic connective tissue. Elastic connective tissue. And I want you guys to Google slides of elastic connective tissue. And I want you to be able to appreciate basically the elastic fibers in there. Next then, let's move down. So here when we move down, you can see page 14 then is muscle tissue. We'll skip muscle tissue because our job is connective tissue first, and then we'll come back and do this. So to skip that, then we'll move down, and we're going to move down here to page 20. So page 20 takes us to connective tissue part 2. So here you have first hyaline cartilage. So here what we have to do, identification, location, functions, right? Make sure you've got all that down. Here then, it's asking you to identify collagen fibers. You cannot see the collagen fibers, so there's no way you can identify them. I won't ask you to identify them. Here, then it asks you to identify the chondrocytes. Chondrocytes you can appreciate right inside of here. So here you can see a chondrocyte. This whole thing here is a lacuna. It's a hollow chamber that gets occupied by a chondrocyte. So the cell, the chondrocyte, is inside of this lacuna. Okay, here you can see the lacuna looks empty. And if a cell comes and occupies inside, then, then you'll see it'll look similar to something like this. So the actual space is the lacuna. The cell inside the space is called the chondrocyte. The chondrocytes, their function is going to be to maintain the matrix of this connective tissue. Now here off to the sides, we can appreciate chondroblasts. Chondroblasts are actually producing the matrix. So once they mature, they occupy these lacunae, and then their job is to maintain the health of the matrix versus producing the matrix. Don't worry about chondroclast. You could cross that off. And the lacuna we got there as well. Next, then here we have elastic cartilage. Same thing. Identification, locations, functions, collagen fibers are not visible here, so you don't have to see. You could cross them off. Elastic fibers, though. Here you can see a whole bunch of elastic fibers. All these fibers, elastic fibers. Elastic fibers. And the same thing here. The space is the lacuna. In the lacuna is the chondrocyte. The space is the lacuna. In the lacuna is the chondrocyte. Here lacuna is empty. No chondrocyte. Here it's occupied. And the blasts are going to be off to the sides. You can see here. And the lacuna you got there. Fibrocartilage, we can see here next. Identification, location, functions, collagen fibers here. That's all these fibers you see in here. Collagen fibers. Collagen fibers. All collagen fibers. All collagen fibers. And what do you have in here? Lacunae, occupied by chondrocytes. Very good. Lacunae occupied by chondrocytes. You can't see chondroblasts here, so don't worry about that, nor chondrocytes. Or chondroclasts, I mean. Class, in none of them, you don't have to see. Then the lacuna you've got there. Next, then we've got bone, osseous tissue. So here, you can see, again, identification, location, functions. And then here you have to be able to identify an osteon. Here's one osteon. Here's another osteon. Here's another osteon. Here's another osteon. Here's another osteon. They're the structural units. Structural units of your compact bone. Okay? So now in relation to this osteon, you can see right at the center, the osteon canal. The osteon canal, central canal, Haversian canal, right at the center of the osteon. It's important because it's going to have blood vessels running through and nerves. All running through right at the center. Next in here you have osteocytes. Osteocytes are going to be in these lacunae. So you can see surrounding the central canal are these lacunae. Look at all these lacunae. Inside the lacunae are going to be your osteocytes. Osteocytes. You can't see the blast and you can't see the class, but you have to know their functions. The blasts are the immature cells. Cytes are the mature cells that they become. And then the class are responsible for breaking down bone. So all these lacunae, 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 lacuna, 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 lacuna. Grab this one, lacuna, 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 lacuna. Okay? Lamellae next. Now, to appreciate the lamellae, what we'll do is we'll zoom in on one osteon. When we zoom in on one osteon, here you can see lamellae are like the trunks, like the tree trunks, basically the rings around the tree trunk. When you want to count the age of the tree, we say, right, count the rings. So here you can see the central canal. Now around here you've got one lamella. then another lamella, another lamella, another lamella, another lamella, another lamella, basically a whole bunch of lamellae just surrounding. You can see that central canal. You'll see it in greater detail in the lecture. So that's what the lamellae are. Next thing you have canaliculi. Canaliculi, you can see right in here, there are these cracks. They connect lacuna to lacuna and eventually to the central canal. They allow nutrients to diffuse through, so that way the cells can be nourished. Canaliculi, these cracks. These little cracks, you see a whole bunch of cracks all running through here, little hair-like cracks. All these little crack-like structures. All right in here, all right in here. Next are Volkmann's canals or perforating canals. So what we'll do is we'll move over here. You can see those in this image. So here you can see they connect basically central canal to central canal. And then you have cancellous bone, spongy bone, trabecular bone. So here you have to identify two things. Red bone marrow. You see red bone marrow all inside of here. All inside of here. All inside of here. All inside of here. All red bone marrow. All red bone marrow. And all this pink stuff you see here is trabeculae. That's all trabeculae. Red bone marrow inside of here. Inside of here. Then here you have a slide of blood. Now here in this slide of blood, you have to, again, connective tissue, identification, functions, plasma. Plasma is all this backdrop, all back here, all plasma. Next thing you have erythrocytes or your red blood cells. Here you can see one erythrocyte. Here you have two, one on top of each other. Here they're stacked on top of each other even further. Red blood cell or erythrocyte, 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 RBC, whatever you want to call it. Then you have your leukocytes or white blood cells. Here's one nice white blood cell, leukocyte. And then last you have your thrombocytes or platelets. Thrombocytes or platelets. They're cell fragments, little pieces. So here they're easy to identify. Next then is going to be the last part here, which takes you into then page 14. So here you've got muscle tissue and nervous tissue and integument. So this is the last day of labs is when you'll look at this. So muscle tissue, identify muscle. The muscle I'll show you skeletal muscle. So you have to be able to identify skeletal muscle. You have to be able to know its locations, where it's found. Relative speed of contraction, you can cross that off for all of them. You don't have to know that for any of them. Control, what type of control is skeletal muscle under? You will see it's under voluntary control. Striations and nuclei, basically you have to identify from the skeletal muscle slide. So here, skeletal muscle. Here are all the striations. All the striations. And then right in here, all the nuclei. Nuclei, 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 nuclei. Right? Nuclei, nuclei. Okay? Next then you have cardiac muscle. So here's cardiac muscle. I'll be able to identify it, know the locations where it's found, obviously within the walls of the heart. The speed of contraction, cross off. Control, make sure you got the control down. It's under involuntary control. And then here you have intercalated discs. Look at these intercalated discs. Don't confuse them with the nuclei, because look at the way the nuclei are found. They're vertical. These are horizontal. Horizontal. In between two cells. More intercalated discs. Another intercalated disc. Okay, and more intercalated disc. In between two cells. So it tells you to get the function down. And then the striations here. Here you can appreciate the striations. So the intercalated discs are going to be basically connecting two cells and allowing for communication in between the two cells. And then nuclei. 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 nuclei, right? Nuclei, 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 intercalated disc, nuclei, nuclei, nuclei. Okay, and then here we've got smooth muscle, identification, location. Don't worry about relative speed of contraction. Control, involuntary control. And here, nuclei is really all you can identify. Nuclei, 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 nuclei. A whole bunch of nuclei everywhere. Nuclei, nuclei. They're all stained purple. Nuclei, 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 nuclei. Let's zoom in on these cells. When we zoom in on the cells, here you can see smooth muscle cells. These are the nuclei there. These are like squamous shaped cells. Smooth muscle cells. So it's pointing to the smooth muscle cells in this slide. Next one we have our giant multipolar neuron. Be able to identify it. And then here you have the soma. The soma is another name for the cell body. So right here is the soma, or the cell body. Inside the soma or the cell body, you have the nucleus. Within the nucleus, then you have the nucleolus. So make sure you got those down and you know their functions. And then you have processes. These processes are radiating basically to and from that cell body. So here you could see the axon, one, thicker process, versus the dendrites, many, thinner, tapering processes. Dendrites, and then one axon. This slide is zooming in, basically just showing us a whole bunch of axons, just a whole bunch of axons. So we'll zoom in, and here you can see a whole bunch of axons. Now these axons, they have myelin on them. And this myelin you see on top of these axons is produced by these Schwann cells. It's produced by these Schwann cells. So their function is to produce myelin. Now in between two Schwann cells, you have an unmyelinated region where it doesn't get myelin. This is called a node of Ranvier. Node of Ranvier. Here you have to regenerate your action potential, your electrical signal. So ions flow across to allow you to regenerate your action potential. That's the function of that node of Ranvier. And then here we have integumentary system. So integumentary system, here you can see you've got epidermis all outside of here. Dermis you can see right down inside of here. And then here we have hypodermis. All hypodermis. So here then you can appreciate skin appendages. Now here we can appreciate some sebaceous glands, some more sebaceous glands, some more sebaceous glands, more sebaceous glands, more. They're found next to the follicles or pores. And here you can see the hair, the pili. Hair, the pili. Okay, same thing here. Same thing we would have here. Same thing in here. Right here and here. And then the hair is going to have a few divisions to it. You've got the root and you have the shaft. The root, okay, is going to be basically this part down here. And the shaft is going to be basically this part right up in here. And we'll talk more again in lecture. We'll see that. Then the follicle. This is all the follicle all around here. All the follicle. This is housing the hair. It houses the pili. It's all the follicle. And then the bulb you have right down in here. Add in bulb. You have bulb right down here. So that's all the lab for this first part. Okay. So this took care of, you can see everything here with Module 1. Now with Module 2, when you open up Module 2, okay, so here what's going to happen is you're going to click on Skeletal. When you click on Skeletal, this will pop open. So here you have Skeletal. Now with Skeletal, what you have to do is, as we progress through, it'll tell you histology, and it'll tell you axial skeleton, fetal skull, then it'll tell you skull bones. So you have to follow through with that yourself by reading in the syllabus. So here is histology. You'll go through everything we've done here before with the histology before. So same exact thing you have in here, nothing different. Go through there, and then you have these 4 fontanelles to identify. And here you've got these 2 images to do that from. Anterior fontanelle, posterior fontanelle, and then here you can see sphenoidal fontanelle and mastoid fontanelle. Okay, so that takes care of basically this whole column here. Next, then here you can see what you have is this next column where it starts with skull bones. Now here in relation to skull bones, what you guys have to do is you have to go through each of these bones. So this is like the second lab. The first lab goes through this first column. The second lab goes through this second column. You'll start here. You're going to start with the cranium. You're going to go through, and you have to be able to identify each of these different parts you have labeled here. So here it says sutural bones and Wormian bones right next to it. That's another name for the sutural bones, Wormian bones. That's why they're telling you the 2 names there. And you have to be able to go through, identify everything here with each of these bones, okay? Just like we do in the lecture. All the way down. All the way down. So this is also the second lab. And then the third lab takes you even further, you can see, will move you through to page 34. So now page 34, you'll do the same thing. Vertebral column, go through, find the annulus fibrosus, nucleus pulposus, hernia, and identify all of these parts there. Then vertebrae. On vertebrae, you have to be able to identify the following on all vertebrae. All of these things you see here. And then specific vertebra, you'll look at cervical vertebrae and the first one, the second one, and then the rest of them. And here's what you have to be able to see with those. Your thoracic vertebra, lumbar vertebra, what you have to see there, not much. Sacrum, and then the coccyx, and then moving down. So this is all third lab. The bony thorax, right, sternum, and the ribs. And then the lab after this is going to take you to page 40. This is where appendicular skeleton begins. So when you have appendicular scheduled, this is where you'll pick up from. So this is with lab 3. And you'll continue all the way through, pectoral girdle, clavicle, scapulae, upper limb, moving all the way through to the radius, the carpals, the metacarpals, and the phalanges. This is where lab 3 ends. And lab 4 picks up then from pelvic girdle. So pelvic girdle, moving all the way through, okay, making your way all the way down, okay, for lab 4, and then taking you through to page 46, going through the whole lower limb, identifying all of these different parts here. Okay, I can ask you on the exam by pointing to any of those parts. Where is this? What's this? What is this? What is this? You have to be able to identify that. Coming all the way through this column, all the way up through the metatarsals, phalanges, and then the joints, taking care of all your joints. Okay, so that concludes then everything for skeletal. So all your skeletal labs. Then we'll take the exam on these, and then we'll move to the next one. Now, next one will be muscular system. So for muscular system, once you come here, click on muscular, then what opens up is what you see here. Here now are all the muscles you need to know. You'll use this list of muscles along with the list that you have in lecture and put the 2 together, and that's all the muscles you'll be responsible for knowing about. Now here, some of them have some of this information written out, but you have to go and find it yourself and identify it yourself. Some of this stuff is not 100% accurate, so I'll tell you just like that. Double check all of that stuff. So you'll go through, you have to know all of these muscles, and with muscles you have to know MAOI. Muscle, M, A, action, O, origin, MAOI, insertion. So here you can see muscle, origin, insertion, and action. So for all the muscles I give to you. Every muscle I give to you, you have to know MAOI. all the way through. So this is basically every single muscle that you have to know. All of them are listed here. That's what you have in this section. This is where Atom kicks in. All Atom is another picture source for me to ask you from. You'd use Atom and use Google. I want you to use both. Okay, I can use pictures from Atom or Google and test you from either of them. So that's what you see here. This is basically your list of muscles. That's what you have here with muscles. Okay, all these muscles you have to know. Then when you click on the next section is going to be nervous. When you click on nervous, this is going to pop up. Now here with nervous, what you have to do is you'll have to go through and you have to identify the pituitary gland. You have to identify the pituitary gland from the whole sheep brain, sagittal sheep brain, and the human brain model, and then from Atom. Okay, the sagittal section. Then, the infundibulum on the whole sheep brain, and then the brain model. Cerebral hemispheres, right and left, everything is checked off. So whatever is checked off, you have to be able to identify from there. Pretty simple. If it's not checked off, like you didn't see the infundibulum on the sagittal because you're not going to be able to see it. Lateral sulcus, on the whole sheep brain, you can't see it. A sagittal, you can't see it. So that's why I don't have it checked off. The brain model, you can see it. So whatever is checked off or whatever you have there, I want you to be able to know. Okay? So same thing here. Okay? You do this on the first lab. Second lab, it tells you to move through. And then third lab, you're going to pick up with the next section. But every day, and this will be third lab, cranial nerves. So cranial nerves now, you can see same thing here, whole sheep brain, brain model, and then Atom. Whatever is checked off, that's where you'll be responsible for knowing. Okay, spinal nerves, be able to identify all of these different nerves, know which one is a dorsal rami, which one's a ventral rami, and what plexuses are going to form. And then here we can see all the different nerves. And then also you'll add in histology every day. 2 slides if you add in every day, it makes it easy to go through. First is the neuromuscular junction, motor end plate. You have to be able to identify the axon terminals, the terminal boutons there, the axon branches, and the muscle fibers. So we can see that here. Here's the actual muscle fibers running through. Okay, here's the axon branches coming through. And then here's the terminal boutons. All in here. All in here. All in here. All in here. Okay. Then multipolar neuron, we've gone through that. Spinal cord, you could go through that. You'll see that we do that in the lecture. And the peripheral nerve, same thing there. And then the Pacinian corpuscle we'll check out in the lab, and then know your axons. So here you can see that the multipolar neuron, spinal cord cross section. Here's a nerve, and a Pacinian corpuscle, and then there's your axons. And then here you've got the eye. So the last day for nervous system is going to include the eye and the ear. So whatever is checked off is what you have to know. From the dissection, we're not doing dissection, but you could look at the dissection pictures I have online and be able to identify stuff from there. And then the eye models, and then Atom, basically pictures. Whatever is checked off is what you'll be responsible for knowing about. Okay, all the way down to the ear model. So everything here, make sure you can identify like we do in the lecture. So that's nervous. And then also here during nervous you have your, oh, not this, sheep brain. Here's your sheep brain. You guys will meet this young man who prepared this for you guys. It's been going around for a long time. He's a nurse up here. So here's the sheep brain, and you can see all the parts that you're responsible for knowing about are identified. So very simple for you. And then from nervous you move to viscera and then cardio. So here when you click viscera then, after sheep brain, viscera, this is what pops up. So for viscera, by the time you get there, you'll know all these cavities. You have to be able to identify all these various cavities, okay, in the cats and also the human. And in membranes, you got to know all these membranes down now, because we're already towards the end of the semester. And the endocrine system, you have to be able to identify these basically glands on the human and then also the cats. And then some histology slides you have there, which we'll go through in the lecture for that section. So here you can see, then you'll have after that digestive system. So whatever day those are scheduled, you'll follow through and go through, check that out. Identify basically these parts on the sagittal cat head, all these parts you have there. And then the whole cat, you've got all these parts here. The parotid gland you can't see on the cat, so these things you can't see on the cat, these are atom only, so human only. So you have to know human, and you're obviously not seeing the cat, so you're knowing everything for human if you're just online. So go through, identify everything here, all these various organs, and then you have some histology. And histology will go through the lecture, and you have that checked off from the lecture also. Nothing complicated there. And these pictures all coincide, just like we've been seeing. And then same thing here, urinary system. So whatever day urinary is scheduled, everything you have to identify urinary. It says cat, but this will be human. Everything is human, since we're online because of COVID. But once we're in class, then you students will have to do everything in cat and have it down basically there in human as well. Cow kidney. I have this cow kidney on my website. You're welcome to use that. You can identify all these parts on the cats as well. I've got all the pictures there for your added bonus. And then histology, we'll have gone through and checked that out in the lecture. Reproductive, everything here with male. And then here you can see slide with male. Next then, we'll move through. You'll have then female, reproductive, go through everything here with female. Everything here. Then you have respiratory. Respiratory, you have your larynx model. I have a picture up on my website you can see there as well. And you can find these models online, and in the lecture we go through them. Sheep larynx, you have there also. Again, for your added bonus on the website. And then the cat, you have to identify these parts there. Human, you'll do there on it for human then. This is all respiratory. So human respiratory, and then slides, histology. And then we're done with viscera. And then last you have here, when you click, will be cardiovascular. So once you click cardiovascular, then what's going to open up will be this section here. Now this section here you can see starts you off on page 128, and you have heart models and sheep hearts. And you'll basically check off from both of those. Whatever's checked off, you'll be responsible for knowing. And then here you have pictures we'll use for all of this stuff. Basically heart as well. So pictures, basically all pictures, know that from there. Same thing here with blood vessels, know them from pictures. And once we get back in the classroom, we'll have models and we can test from the models. But here we have pictures. So same thing here, know all your veins, go through every single one of these veins. And make sure you got all these veins down in relation to the human. And then blood, you have histology here, blood smear, your erythrocytes, platelets, plasma, leukocytes, your white blood cells. By the end, you're going to know every single one of these. So you'll go through those. And then a vein, an artery, you can see in here. So each and every one of those blood cells. And then you can see a vein and an artery there. So you have to be able to identify those. And then here, same thing, artery, vein, going through those. And then going through the arteries. So identifying arteries throughout the whole body there. Checking all of those out. So that's how you'll guide yourself through this free lab I provide you guys. Awesome.